Hello class, a very warm good morning to everyone. Myself Vivek Prakash Yadav and I am here to take your biology class for class 10. So in the last class we are discussing about introduction of reproduction. In that we have discussed about the definition of sexual and asexual reproduction, and in that we have discussed about the type of asexual reproduction. So we are again going to start from there. So what are the type of asexual reproduction? Types of asexual reproduction. Okay. Type of asexual reproduction. I told you that asexual reproduction is also different types. Now, on what basis organisms have adapted the particular types of particular mode of asexual reproduction? I told you in the last class that in the case of organism that following asexual reproduction, there is also difference in their body organization. Some are cellular, unicellular, some are the combination of cells, and later on again we move little higher, they will have the tissue level organization and so on. So depending upon their complexity also, they are classified into different types, or they have adapted different mode of asexual reproduction. So here we are going to start what are the different types first, then we will discuss separately one by one each. Okay. So first of all, that is simple, that is fission. Okay, fission. Okay, now we will discuss about fission. What is fission? Fission, it is a mode or type of, we can say, asexual reproduction. Asexual reproduction. Okay, it is a type of or mode of asexual reproduction in which parent body is divided into two equal half. Okay, parent body is divided into two equal half. This is the important thing when we are going to talk about fission. Okay, now actually fission is also two types. Fission is two types. Okay, it is two types. Okay, fission is how many types? It is two types. First one is binary fission. Binary fission, and another one is your multiple fission. Okay, one is your binary fission and another one is your multiple fission. Now what happened in binary fission, as its name indicating, by means what I said here, that in this parent body, okay, parent body divide in two, parent body divide into equal half. Okay, parent body divide into two equal half. We can just take the example of amoeba. Okay, example of amoeba. Now what happened? We all know that amoeba does not have any particular symmetry. Yes, it doesn't have any particular symmetry or shape. So it can be divided from any plane in equal half. The matter is that here the organism should be going to be divided into equal half. Okay, but now we are taking example amoeba. So in case of amoeba, the body can be going to be divided. Okay, the body can, this is nucleus, this is eukaryotic. So what will be happen here? The body is going to be elongate. Okay, with that the genetic material, that is nucleus. Okay, nucleus is also going to be elongate. And what you will see that the nucleus, okay, going to be divided into daughter cell. Okay, going to be divided into daughter cell. This is what binary fission. This is what binary fission. That parent body, when undergoes the process of binary fission, the nucleus is going to be elongate, and after elongation, the pinching off will take place, and finally, from the parent, two daughter cell form. Two daughter cell form. Okay, two daughter cell form. This is binary fission. Now, in case of binary fission also, okay, in case of binary fission also, there are some features we need to be considered. Hope you people are, we will discuss multiple fission later. First, we will discuss binary fission completely. Now, what I told you that in the case of amoeba, the binary fission was take place in asymmetry, means it can be going to be divided into equal half in any plane. But when there are some organisms, their body cannot be divided into equal half at any plane. They are a particular symmetry. Okay, they have a particular symmetry. Only in that symmetry, only in that plane, they are going to be divided into equal half. You can take the example of paramecium. Okay, example you can take paramecium, leishmania. Okay, these are examples of protozoans. These are the examples of protozoans. Now, if you see the structure of paramecium, you find something like this structure. Okay, sleeper cell. Okay, something like this. Now, if we need to be divide this into equal half, okay, if we need to be, this is cilia. Okay. What I am saying, if we are going to divide this, okay, into equal half, if we are going to divide them in this way, that is not going to be divided in equal half. Okay, that is not going to be divided in equal half. So generally, in case of paramecium also, this pattern of divisions going to be take place. That is longitudinal division. And the best example you can see here in the case of leishmania. In the case of leishmania, okay, when you are going to see the structure of leishmania, it is something like a long flagella is there, and okay, finally this is the shape, okay, of leishmania, okay, protozoans. So in that case you cannot do transverse division. Transverse division not going to be divide them in equal half. So what happens? There is need of longitudinal binary fission. What? Longitudinal binary fission. So what we have discussed here, what is fission? Fission is a type of asexual reproduction generally takes place in the case of eukaryotic or unicellular, in which the parent body divided into two equal half. Now, type of binary fission we have discussed about type of fission we have discussed about two types, that is binary fission and multiple fission. In case of multiple binary fission, the condition is that we have to concern or the animal body organization or symmetry. The symmetry of organism also decide the pattern of fission. Okay, in case of binary fission, the symmetry of organism going to be decide the which type of binary fission going to be take place. Suppose I told you, in case of amoeba, from any plane they can be going to be divided in equal half. But when we are going to talk about paramecium or leishmania, in that case there is a need of longitudinal division. What longitudinal division? Because what is leishmania? It is a type of protozoans which cause kala ajar. Okay, it is a type of protozoans which cause kala ajar. Now their body have a whip-like structure, that is flagella. Okay, the body of leishmania have flagella. So if transverse division is going to be performed, then there is no equal division. There is no equal division of the organism going to be take place. As a result, they follow longitudinal binary fission. They follow longitudinal binary fission. So hope you people clear with the binary fission. Now what next? what next is multiple fission. Okay, multiple fission. Now, in case of multiple fission, see, it is actually performed during adverse condition. We can say during unfavorable condition or in adverse condition, what happened? There are organisms which going to follow multiple fission. The best example for this, okay, the best example we can see here for the multiple fission is plasmodium. Okay, plasmodium. What is plasmodium? Plasmodium is also a type of protozoan which is the causative agent of malaria. Okay, which is the causative agent of malaria. It is the species genus of the malaria causing organism, plasmodium. So, in case of plasmodium, what happens during adverse condition? Okay, during adverse or unfavorable condition, what they do? The cell inside which there is presence of nucleus. Okay, inside which there is presence of nucleus and the cytoplasm. Yes, the cell, the cytoplasm, and in that there is nucleus is present. So what happens during unfavorable condition? They call cyst formation or spore formation. Okay, they form. What they do? They develop a spore. Okay, they develop a spore or covering which protect them in unfavorable condition. Okay, that spore protect them against that unfavorable condition. But internally, what happens? The cell is active, and the nucleus, okay, the nucleus which is going to be divided into small nuclei. Okay, the nucleus which is present inside the cytoplasm. Okay, inside the cytoplasm, they are going to be divided into small nuclei. Okay, they are going to be divided into small nuclei. Now what happened? This nuclei going to take a few amount of the cytoplasm. Okay, each nuclei which is divided. Okay, each nuclei which is divided. Okay, they uptake few amount of the cytoplasm around them, and this inside the spore. Okay, inside the spore or membrane structure, inside the plasma membrane, and then this is covered by the spore. Now they are remain in such condition. When favorable condition come, okay, when favorable condition come, see, plasmodium is also flagellated. So when favorable condition come, what happen? The spore get ruptured. Okay, the spore. What happen when the favorable condition come? The spore gets ruptured, and all this small plasmodium, okay, means all the small nuclei, which is by the multiple fission. This is what multiple fission, okay, and many small daughter nuclei, you can say, or daughter plasmodium, okay, they release from the inside the spore. Okay, so this is the example of multiple fission. In case of multiple fission, the organism which is undergoes actually multiple fission performed during unfavorable condition, and the nucleus is getting divided into small nuclei. The nucleus is getting divided into many small nuclei, and each nuclei take a few part or few percentage of the cytoplasmic material around them. And when favorable condition come, okay, when the favorable condition come, those small nuclei by the rupture of the spore, okay, spore layer or cyst wall, they all release into the environment or in the particular media. Generally, it is water. Okay. So hope you all are clear with the first type of asexual reproduction, that is fission. Okay, that is fission. Now the second method in which we are going to discuss is. Second type that we are going to discuss in the case of asexual reproduction is fragmentation. Okay, fission, then fragmentation. Before fragmentation, it will be better if we are going to discuss about budding. Now budding is also a type of asexual mode of reproduction which is going to be takes place in the simple organism. They have cellular level of organization, or even we can say tissue level of organization. The best example you can see here is one is your yeast, okay, one is your yeast and another one is your hydra. Okay, the best example you have seen here is yeast and another one is hydra. Okay, now yeast is the example of unicellular eukaryote. Yes, it is example of unicellular eukaryote which follow budding, and in the case of hydra which is multicellular and they have tissue grade organization. Now what happened in the case of yeast? Suppose this is the structure of the yeast inside the nucleus. What happened in the parent body? Okay, in the parent body there is a small outgrowth. There is a small outgrowth, and that outgrowth include or have the few percentage of the nucleus. Few percentage of the nucleus. We can say that suppose this is the structure, and now what happens? Small outgrowth like this was there, in which the small amount of the nucleus was there. Later on what happened? This structure is going to be developed, and they are almost like their parental structure with having the nucleus also. And finally, this structure going to be detached. Okay, the small outgrowth. Okay, small outgrowth that was known as bud getting detached from the parent and survive as individual. Okay, independent individual. So this is the case of budding in the yeast. Now in the case of hydra, okay, in the case of hydra, they also follow the method of budding. In that, what happened? We know hydra have the tentacles, okay, tentacles, a particular structure they have, and okay. Okay, this kind of structure is there in the hydra. What happened when this structure, then this parent organism get matured. Okay, when they are at their maturity, there is a small outgrowth in their body part. Okay, there is a small outgrowth, or we can say accumulation of cell. Okay, due to the cell division, they get aggregated in a particular region. And what happened? This small aggregation or a bud which is formed, they start developing and growing. They start developing and growing, and slowly this bud-like structure going to be increased, and a time will come in which you can see that particular growth. Okay, that outgrowth was resemble like the parent. Okay, they resemble same like the parent. The difference is only that the outgrowth is small in size, but fully they are going to be identical like the parent. And after complete growth, what happened? After complete growth, this outgrowth, this bud is going to be detached from the parent, and they have all the features of the parent. Okay, they have all the features of the parent, and they are going to survive as new individual. It depends upon the condition. They are going to attach to the substratum, then they grow, develop, and again they mature. They also follow the same condition. So first we have discussed about fission. Second we have discussed about second type of asexual reproduction that this is discussed is about budding. What happened in budding? In case of budding, what happened? There is a small outgrowth from the parent body, and the small outgrowth develop and get matured like the parents. They have all the structure of the parent, and when they fully develop, they get detached from the parent and survive as individual. Okay, they are going to survive as independent individuals. So in today's class, we have completed two types of asexual reproduction. We have dealt with the definition of the asexual mode of reproduction, and two types, that is fission and budding. In fission, we have discussed about multiple binary fission and multiple fission. In case of binary fission, we have discussed about the consequences that if asymmetrical organism is there, then from any plane it can be divided into two equal halves. But if there is a particular symmetry, then they have to be followed according to that so that they get divided into two equal half. We have discussed the example of paramecium and leishmania, and the another type that we have discussed is budding. In case of unicellular, that is yeast. Okay, small bud will develop from the parent body, and that bud contains the percentage of nucleus, few percentage of nucleus, and when they grow and mature, the bud get detached from the parent body and survive as independent individual. In the case of hydra also we have discussed and understood the concept. So hope you people understood here fission and budding. In the next class we will discuss about fragmentation, regeneration, and so on, whatever the next topic we are remained with. And with that, thank you and have a nice day.